Okay, we're starting recording. So we'll go back to the PowerPoint. So carrier mediated transport, meaning that you have a special membrane protein to move a substance. So in the kidney, you may have special channels that move water in and out of the cell. Active transport depends upon energy from ATP, and an example is the ion pump that moves sodium, potassium, sodium out of the cell, potassium into the cell. So what's facilitated diffusion? You have a special protein carrier that moves certain substances into the cell. So that's facilitated diffusion. It can be stopped by changing the shape of the protein. So facilitated diffusion means that a special molecule is dedicated to carrying in a special substance. The sodium potassium pump, as I said, moves potassium in and moves sodium out, and it takes energy. Anytime you see ATP being converted to ADP, you know that energy is being released. So that's active transport. This protein has to have energy because it's pumping sodium to a higher concentration of sodium. It's pumping potassium to a higher concentration. So it takes energy to force this potassium to an area where the potassium concentration is high. It takes energy to move sodium from a low concentration of sodium to a high concentration. Okay, just know what endocytosis is and exocytosis. Okay, sometimes special vesicles form that will bind only to certain molecules. That's receptor mediated endocytosis. So, cell eating, when cells form a little circular, spherical container, uh, the receptors here help control exactly what is brought into the cell by the formation of this spherical package. Read through this and just know in general what endocytosis is. Exocytosis, movement into the cell, movement out of the cell. Another term for bringing substances into the cell is called phagocytosis. It means cell eating. So the cell forms a special little pocket that forms a vesicle, and this could be a bacteria. Lysosomes bring enzymes to break up the bacteria. The bacteria is broken up and then released into the extracellular environment. So white blood cells that consume bacteria, this is phagocytosis. The stuff inside the cell is cytoplasm. It includes the cell membrane, nucleus. The cytosol is the cytoplasm, and within that cytoplasm, there are organelles such as mitochondria, nuclei, endoplasmic reticulum. So make sure you read through the list of organelles and make sure that you know what each one does. The cytoplasm is the cell, is the fluid in the cell. Materials dissolved there, ions are in there, soluble proteins, structural proteins, and other proteins or other chemical classes that are dissolved in the cytoplasm. It's very important to know that there's a high concentration of potassium inside the cell versus a high concentration of sodium outside the cell. That's the way cells are set up to create an electrochemical gradient. So based upon the charge of potassium and sodium, you can vary their concentration to change the electrical properties of the cell and to change the overall ability of the cells to hold a charge, meaning that the inside is more negative than the out, and to help the cell control what comes in and what goes out, helps the cell generate nerve spikes, muscle contraction, and so on. So what are the organelles? Read through this list, all this list of stuff. So it's your job to read the list and make sure you know what they do. So these are all various mitochondria, endoplasmic reticulum, secretory vesicles. So you should know these organelles, little organs in cells, and just in general to know what they do. So here's tells you what centrioles do: microvilli, cilia, flagella, ribosomes, proteasomes. Tells you what endoplasmic reticulum does. It's two types, rough and smooth. The endoplasmic reticulum is a connected network of tubes. Rough endoplasmic reticulum has ribosomes, which are involved in protein synthesis. Ribosome is the site where proteins take amino acids, join them together, and to make proteins. Okay, here's the endoplasmic reticulum, the smooth endoplasmic reticulum. No ribosomes on the smooth endoplasmic reticulum. Know what Golgi apparatus does? It's involved in packaging, putting substances in secretory vesicles. It also deals with packets of enzymes. So you see that the Golgi apparatus can bud or release these little vesicles that contain certain substances. And know that endoplasmic reticulum takes the proteins made at the ribosome, puts them into vesicles. They're processed and stored and then released in the cell. So if you are the pancreas and you're making insulin, insulin is a protein made in the endoplasmic reticulum. It will be packaged in these secretory vesicles. When you eat a meal of very high protein, that insulin protein is released and carried in a vesicle. Know what lysosomes do? Cells are in a particular environment based upon the size, charge, solubility of a substance. All those allow the cell to decide what goes into the cell and what goes out of the cell. So cells can change their internal structure and their functions over time. Know what mitochondria do? They're the power source, the source of glucose for sugar. Sugar is used to make more ATP. So glucose is eaten, it goes into the mitochondria, and most ATP is produced by the mitochondria, and the cell uses ATP by releasing energy by taking off a phosphate. And so we have the ability of a cell to take glucose and to make ATP. So the mitochondria have this little bean appearance. They are thought to be bacteria that invaded the cell, that invaded larger cells, and the bacteria liked it inside the cell, so they stayed. That's called endosymbiosis. Mitochondria use oxygen. They generate carbon dioxide and ATP. The nucleus is another organelle. It's where DNA is located. It controls cellular operations when the cell copies the directions from DNA to RNA, and that RNA goes into the cytoplasm with instructions. Tell the cell, make this protein, make this lipid, get rid of this substance. So all of the compounds that are in the nucleus, which is DNA, the DNA stays in the nucleus, but messenger RNA is the messenger which goes all over the cell, telling the cell what to do. Make a protein, break up this protein, make a lipid. Okay, so read through this. That's what the nucleus looks like. It has a structure called the nucleolus. Chromatin is condensed DNA. There are pores that are in the nuclear envelope or nuclear membrane, and those pores allow substances to go in and out of cells, in and out of the nucleus, sorry. We all have 23 pairs of chromosomes, and we're getting toward the end here. Let's stop this here and have a small stop recording.